मॉर्निंग स्टूडेंट्स, वेलकम टू माय चैनल नर्चरिंग इंटेलेक्ट। अगर आपने मेरा चैनल अभी तक सब्सक्राइब नहीं किया है तो डोंट फॉरगेट टू सब्सक्राइब द चैनल एंड इफ यू लाइक द वीडियो hit the like button, comment and share.
So today under the chapter Biotechnology Principles and Processes, we are going to study another important tool used in recombinant DNA technology, that is cloning vector. In this lesson, we are going to study about the cloning vector which is used for bacteria. So let's begin. What are cloning vectors? So cloning vectors, they are DNA molecules in which we can integrate a gene of interest. And later on it can be used to replicate inside the host cell. In bacteria, the commonly used cloning vectors are plasmids and bacteriophages. What are plasmids? As you studied already, in bacteria, they have their own chromosomal DNA. In addition to that, some bacteria, they have a circular extra chromosomal DNA. This DNA is capable of replicating on its own because it has its own origin of replication. So such autonomously replicating extra chromosomal DNA are known as plasmids. Some plasmids, they may have 1, 2 or 3 copies per cell. But there are others which have high copy number and they maintain up to 100 copies per cell. For recombinant DNA technology, we make use of plasmids which have high copy number, so that when we introduce our gene of interest in it, that gene of interest will multiply to the number equal to the plasmid inside the bacterial cell.
The second important cloning vector for bacteria are bacteriophages.
Bacteriophages are the viruses that attack bacteria. They have double-stranded DNA as the genetic material. So when they attack bacteria, they inject their DNA in their cell. Inside the cell, the bacteriophage viral DNA, it replicates and forms multiple copies. So there are two reasons why we select bacteriophages as the cloning vector. First, they have high copy number per cell, and second, we can integrate large DNA fragments in their genome. So due to these two reasons, bacteriophages are also preferred cloning vectors used for prokaryotes. We have already studied that in recombinant DNA technology, whatever vector we are using, first we treat that vector with a restriction enzyme. Same enzyme we are using for the DNA from which we have to isolate our gene of interest. Then mixing the two and adding ligase will help in integrating the foreign gene into the vector DNA. The DNA so obtained is known as recombinant DNA. So this is recombinant DNA in which plasmid DNA is there, which is acting as vector, and our gene of interest is ligated. This recombinant DNA is used to transform the bacterial cell. They are taken up by the bacterial cell when we add them in the bacterial culture. And the process is called transformation. Once the transformation occurs, according to the type of origin which they have, they multiply inside the bacteria to form their coded copy numbers. As well as this gene of interest is also going to multiply. So this is known as gene cloning. In E.coli, the plasmid which is used as cloning vector is PBR322. PBR322 is the first genetically engineered plasmid vector which is widely used in recombinant DNA technology. In normal E. coli, this plasmid is not present. It is artificially constructed by two scientists named as Bolivar and Rodriguez in the year 1972. So accordingly, the nomenclature is done. P stands for plasmid. Here BR stands for the name of the scientist who constructed it, that is Bolivar and Rodriguez, and the number 322. It is given to distinguish this plasmid from other plasmids developed in the same laboratory. Why this is an ideal plasmid? Why we are not using the natural plasmids which are found in bacteria? Because there are certain features which these scientists introduced in this plasmid which make it more suitable to be used in recombinant DNA technology.
So let's study the features required for cloning into a vector.
First thing, a vector must have an origin of replication. Then only the integrated DNA will be able to replicate inside the host cell. Second, selectable markers. So selectable markers, these are the antibiotic resistant gene present in the vector. They help in selection of transformants over non-transformants. Third is cloning sites. So cloning sites are the sites which are recognized by the restriction enzyme. First they recognize these sites and then they cut the DNA in this particular region. As they cut the DNA, now the sticky ends are generated and we can introduce the foreign DNA into it. So these are the three important features needed in any vector: origin of replication, selectable marker, and cloning site. Let's study about origin of replication. So what is origin of replication? It is a sequence of nucleotide which is responsible for initiation of replication. So any foreign piece of DNA, when you link with the origin, okay, if it is linked to an origin, we can make it replicate inside the host cell. That's why if a naked DNA just enters into the cell, it won't be able to multiply because it doesn't carry the origin of replication. This origin of replication in PBR322 is derived from E. coli plasmid, which is naturally present. Second thing, DNA holds the number of copies of the recombinant DNA in the host cell. So in recombinant DNA technology, we use the vector which supports high copy number so that many times our gene of interest will get multiplied. Selectable is selectable marker. Selectable markers are the genes which help us to select transformants and eliminate the non-transformants. There are two selectable markers present in PBR322 and they are tetracycline resistant gene. This gene offers resistance against the antibiotic tetracycline. And the second one is ampicillin resistant gene. So it produces chemicals which provide resistance to the bacteria against ampicillin. In normal E. coli cell, if they have plasmid, these plasmids, they do not carry these resistant genes. That's why we are using PBR322. Once we develop the recombinant DNA, recombinant DNA क्या है? जिसमें हमने foreign gene को introduce कर दिया है. That vector is known as recombinant DNA. So then we mix the recombinant DNA with the bacterial culture, and some of the bacteria they pick up the DNA and some will not pick up the DNA. Those which will pick up the DNA, foreign DNA, they are called transformants, and those which will not pick up the DNA, they are called non-transformants. So since the transformants are having the recombinant DNA, okay, they are resistant to antibiotics, whereas non-transformants, they are not resistant to any of the antibiotic. So if we grow them in a medium containing antibiotic, the transformants will survive and the non-transformants will die. So in this way, selectable markers, they help us in selection of transformants over non-transformants. In a clone, third unique feature which is needed in a cloning vector, that is cloning sites or unique restriction sites. Cloning sites, these are the sequences where specific restriction enzymes can make a cut so that our gene of interest can be incorporated into the vector. So in PBR322, in the ampicillin resistant gene, we are having two restriction sites or cloning sites, that is PVU1 and PST1. Whereas in tetracycline resistant gene, there are two restriction sites for enzyme BAM-H1 and SAL1. Okay, so cloning sites are very important because it is the place where we introduce our gene of interest. Be kept. 2 points must be kept in mind while selecting a vector. Okay, the vector should have a few or at least one recognition site, because then only we will be able to introduce the foreign DNA in it. If a vector is having more than one recognition site, then also it will be a problem, because the restriction endonuclease will cut the vector DNA into many fragments, and that will complicate the gene cloning procedure. Now, ligation of alien DNA is carried out at these restriction sites or cloning sites which are present in one of the two antibiotic resistant genes. So we always introduce the DNA, foreign DNA, into one of the antibiotic resistant gene, cloning site present in one of the two antibiotic resistant gene DNA, which take up the recombinant DNA. They are known as recombinants. Now the next task in front of us is to select the recombinants and separate them out from the non-recombinants. So what are non-recombinants? Here you can see in the picture, this is the vector plasmid, and we already opened this plasmid using a restriction enzyme. Now if the foreign gene get introduced in it, so the DNA formed will be recombinant DNA. But sometimes what happens, instead of the gene getting integrated with it, the two sticky ends, they join on themselves. So again the intact plasmid is obtained. So that plasmid is not carrying the gene of interest. So it is known as non-recombinant. Now in the solution where we developed recombinant DNA, we will have several recombinant DNA molecules and several non-recombinants where the gene of interest is not there. Bacterial cell can take up both non-recombinant and recombinant. So those cells are called transformants. So it is easy to select the transformants, but the next level of difficulty comes where among the transformants we have to select the one which are carrying the gene of interest, that is recombinant DNA. So how we select recombinants? The first method is insertional inactivation of antibiotic resistant gene. So let's see what's that. Used for E.coli. Okay, this is the plasmid PBR322 used for E.coli. Now, this is the foreign gene which we want to introduce in E.coli, this vector plasmid, vector PBR322. And suppose the cloning site which we choose is BAM-H1. So we treat this vector with BAM-H1 restriction enzyme. So it cuts the DNA in, the vector DNA in tetracycline resistant gene region. The same enzyme was used to isolate the gene of interest from the rest of the DNA. Now when we bring them together and add ligase, the gene of interest gets introduced in the middle of the tetracycline resistant gene. So this plasmid is now known as recombinant plasmid, and it has lost its tetracycline resistant property. It no longer provides tetracycline resistance to the bacteria which will carry this plasmid. Okay, so all the recombinant plasmid, they will lose tetracycline resistance due to the insertion of foreign DNA in it. Okay, that's why it is called insertional inactivation. Produce the chemical which provides protection to the bacteria against the antibiotic tetracycline, but its ampicillin resistant gene is still intact. So recombinant bacteria, they are resistant against an ampicillin antibiotic but not against tetracycline. It is known as DNA becomes the part of the vector PBR322. It is known as recombinant DNA or plasmid. The transformation of E. coli by recombinant plasmid. So as already discussed, when we treat the vector and the foreign DNA with restriction enzyme, sticky ends develop. Okay, and when we add ligase to the solution, the one probability is there that the gene will get integrated with the vector DNA, and this plasmid is called recombinant plasmid. The second probability is that instead of incorporating the foreign gene, the two sticky ends, they again join, okay, which is done by ligase. So these are the non-recombinant plasmids. So in the solution we have non-recombinant plasmid, we have recombinant plasmid. So to this solution we add bacterial cells, that is E. coli, and providing appropriate condition, the E. coli bacteria will take up these plasmids. Some of the bacteria, they pick up the non-recombinant plasmid. Some of the bacteria, they will pick up the recombinant plasmids, and some bacteria, they will not pick up any plasmid. Okay, so those which are not picking up any plasmid, they are called non-transformants, and the one which pick up the either the non-recombinant or recombinant plasmids, they are called transformants. So when we introduce the plasmid into bacteria, we obtain three types of cells. Okay, so in this culture, see, this whole procedure is carried out in the liquid medium. Okay, so we have three types of cells: non-recombinant cells, which are having only the plasmid, gene of interest is not there; recombinant cells, which are having the gene of interest, that is recombinant plasmid; and non-transformants, which do not take up any DNA from the external medium. Okay? Now we have to select among these three types of cells which are present in the culture. We want to grow only the recombinants because they carry the gene of interest, and we are interested in the product which is produced by this gene. Okay, so how to select these recombinants? Let's see. For the bacteria culture, liquid culture, we transfer the bacteria on a petri dish in which agar that gel is present. Okay, nutrient medium in the form of gel is present. So we transfer the colony. In the colonies there are three types of bacterial cells present. This one, the blue one, you can see these are the non-transformants. They have no plasmid. So they are not resistant either to tetracycline or ampicillin because इनमें plasmid ही नहीं है, तो दोनों antibiotic इसको kill कर सकती है. Then second one are the transformants with non-recombinant plasmid. So इसमें क्योंकि foreign gene introduce नहीं की है तो tetracycline gene intact है, so they have resistance to both tetracycline and ampicillin. And the third type of cells which are present in the master plate, they are recombinants. Okay, transformants with recombinant plasmid. So they have only resistance to ampicillin, as due to the insertion of foreign gene into the tetracycline resistant gene, it has lost its property of resistance to antibiotic tetracycline. Okay. So from the culture, we transferred the bacteria on the gel medium, and we get 3 type of colonies of bacteria on the Petri plate. Now, first of all, we select transformants over non-transformants. So here हमने देखा master plate में we were having 3 type of cells. This one non-transformant. These are the 2 transformants. One is having recombinant DNA, the other one is having non-recombinant plasmid. Okay, so transformed cells can be separated out, easily selected from non-transformant by growing them in ampicillin containing nutrient medium. So we prepare another Petri dish in which in the nutrient medium we add antibiotic ampicillin. Since the non-recombinants, they do not have the plasmid, so they will die in the ampicillin medium. The colonies of bacteria which grow, they are the transformants. Now, among these transformant, one is non-recombinant, another is recombinant having our gene of interest. So, how to select them? Okay, for this we perform replica plating for selection of recombinants over non-recombinants. So here the red dots which are there, these are the non-recombinants, these are the recombinants, and this is our master plate. Okay, so we take a muslin cloth or a velvet cloth and we press that velvet cloth over the master plate. The bacterial colonies, they get transferred on the velvet cloth. Okay, and then we prepare 2 other secondary plates. Okay, one of the plate is containing antibiotic tetracycline, the other one is containing antibiotic ampicillin. Okay, and we just press this over these 2 plates. So all these colonies in the same position, they will get transferred in these 2 secondary plates. Okay, now what will happen? Non-recombinants. Okay, non-recombinants, foreign gene नहीं है, इसमें ampicillin resistant gene भी intact है, tetracycline भी है. तो ये जो non-recombinants हैं, they are growing in both the plates. आपके tetracycline medium में भी grow करेंगे and ampicillin medium में भी grow करेंगे. Recombinants, their tetracycline resistant gene is inactivated due to the introduction of foreign gene. So they are no longer resistant to antibiotic tetracycline. So these green colored colonies of recombinants, they die in this plate, but they will grow in the medium containing ampicillin because they have ampicillin resistance. Okay, so now what we do? We came to know these are the colonies. So in the same location in the master plate, we look for the colonies and we pick up the recombinants and culture them, subculture them. So in this nutrient medium, we will be having pure recombinant colonies and they can be further cultured on large scale as per the demand. So this is how insertional inactivation of antibiotic resistant gene helps in selection of recombinants over non-recombinants. Okay, but this procedure is very cumbersome because we have to simultaneously plate them on 2 different plates having 2 different antibiotics. So there is an alternative selectable marker method which is developed. Let's see what it is. They differentiate recombinants from non-recombinants on the basis of their ability to produce color in the presence of a chromogenic substrate. What are chromogenic substrates? Suppose this is the substrate, chromogenic substrate. So when it comes in contact with a proteolytic enzyme, okay, the enzyme converts it into a product that product is colored one. ठीक है, ये colored product क्या होगा? When it will precipitate on the cell membrane of the bacteria. So the bacterias, they will form colored colonies. Okay. So in the plasmid PBr322, there is LAC-Z gene present. Now in this case, we are not introducing our foreign gene in the antibiotic resistant gene area, but we introduce the gene in this LAC-Z gene. So this LAC-Z gene is producing an enzyme beta-galactosidase. Beta-galactosidase, it converts a chromogenic substrate X-gal into a blue colored product. So when we introduce this plasmid, so this is a non-recombinant plasmid right now, okay, not having any foreign gene. When this enters into the bacterial cell, that is the transformation is occurring, okay. Due to this reaction, due to the formation of colored product, these bacteria will form colored colonies, blue colored colonies. So this is the first case where the transformation of bacteria is taking place by non-recombinant plasmid. Okay, so non-recombinants are going to form blue colored colonies.
Now let's see the second step.
Used our gene of interest in this, in between the LAC-Z gene. So the gene becomes non-functional, it gets inactivated. Now it no longer produces the enzyme beta-galactosidase. And thus the substrate which is added is not getting converted into product. And the color of the substrate is colorless. When the bacterial cells, they pick up the recombinant plasmid, okay. So such bacterial cells, they will form white colored colonies. Here the non-recombinant plasmarios, here the non-recombinant plasmid transforms the bacteria. So such colonies are blue in color, whereas when recombinant plasmid transforms the bacteria, those bacteria will form white colored colonies. So we can pick up the recombinant white colored colonies from the Petri plate, and we can subculture it to grow the recombinants on large scale. So this is how this alternate method of selection of recombinants makes our work easier. So let's recap.
So let's recapitulate what we have studied in today's lesson.
Vector, the features of the cloning vector, how we can select recombinants over non-recombinants. In this we studied two techniques, one by insertional inactivation of antibiotic resistant gene, and second by using alternate selectable markers, which are used for study about the vectors which are used for transformation of plant and animal cells, and we will also study the different methods used for transformation. If you like the video, please hit the like button, comment, share, and do not forget to subscribe my channel. Thank you everyone.